Hello and welcome to Biology Interest. Today we're looking at producing DNA fragments as part of the A-level revision series. Over recent years, technology has been developed to help isolate genes, clone these genes, and then transfer them into microorganisms. These microorganisms can then produce vast quantities of the desired protein, which can then be used for various things and to help try and cure various illnesses. Now, what we're going to be looking at today in this video is the ways in which DNA fragments of the desired gene, so the first step, can be created on the first step of producing desired proteins. And then I'll do other videos going along the series of things you do to take this fragment of DNA into vast quantities of DNA and vast quantities of protein. Now, there are three ways we have to know about for A-level biology. That's using reverse transcriptase, using restriction endonucleases, and also using a gene machine to help produce these DNA fragments. First of all, we're going to look at using reverse transcriptase. Now, with this method, you need to take a cell that produces lots of mRNA for the protein that you want to produce, so it produces lots of mRNA. For example, the B cell in the pancreas, which produces lots of mRNA that codes for the protein insulin. So if you wanted to produce insulin, we'd take this cell and we'd remove the mRNA from it before it gets translated. Now, you need to take this mRNA and take a single-stranded copy of the DNA called complementary DNA using reverse transcriptase. So essentially, the reverse transcriptase would bind with the mRNA, and instead of producing another mRNA strand, it would produce a complementary DNA strand. Now, this single-stranded DNA can then be isolated, so taken off the mRNA, and as it's single-stranded, we need it to be double-stranded. It is then converted to a double-stranded piece of DNA using the enzyme DNA polymerase. Now you have a copy of a human gene insulin ready for insertion into a plasmid or something similar like that. Now, while this method is perfectly doable, it does have the inherent problem that mRNA is extremely unstable, and so it's quite hard to keep it around long enough and stable enough for you to produce a complementary DNA strand from it. So really, you don't want to be doing this if you have a choice. So the next method, which is more commonly used, is the method by restriction endonucleases. Now, restriction endonucleases are enzymes which cut up double-stranded DNA. Now, it does this by binding with a section of bases in the DNA called a recognition site. There are two ways this restriction endonuclease can cut the DNA. You can either cut it by creating blunt ends, where it cuts straight down the middle of the DNA. This isn't as good because it means it's harder to stick this DNA fragment into a plasmid or something like that. But other restriction endonucleases cut in a staggered way, almost in a zigzag, leaving uneven or sticky ends with unpaired bases on the DNA strand. Now, this is useful because it means you can use the same restriction enzyme to cut this DNA fragment out of a sequence and also cut open a plasmid. And because the two ends are complementary, you can then insert this DNA fragment into a plasmid much more easily when you get to this later, which I'll talk about in another video. And so that's good. And that means it's better than using reverse transcriptase. And it's also better because it uses DNA, which is more stable and so easier to cut. And it's also had its introns removed by that point. So the third step, which you need to know a bit less about, but is a gene machine essentially. It's basically just using a computer to create the genes in a lab where you input the bases you want to use in the desired sequence to create a gene. And it also means the gene has no introns, and then you basically just get the computer to produce it, and you can either then increase its size using the polymerase chain reaction, which I'll talk about later, or in the same way insert it into a plasmid and have a living cell produce vast quantities of this protein. So I hope you like this overview of how you would produce DNA fragments for the A-level biology syllabus. Stay tuned for more videos about biology and random facts about biology from Biology Interest. Thank you.